Welcome back to Pearson Bio Academy. If you are new to my channel, this is Fazna Imran, and this is the place where you can top up your biology knowledge with our short and clear past paper video series. And today we are going to step into past paper questions in chromatography. But before that, if you haven't seen my previous video, check the description box. There I explain how to perform chromatography, what you need, and everything about it. Now, in this session, look at this picture. This is after a paper chromatography has been done. From chromatography, we take readings. First, the distance moved by the solvent front, then the distance traveled by each pigment. In this picture, you can see pigment 1 and pigment 2. So these are the readings we take from the paper. Next comes an important equation, the RF value. RF equals distance traveled by solute over distance traveled by solvent. So from paper chromatography, you take the readings, apply the formula, and you get the RF value. Now my question is, why do we need this RF value? This is simply because it helps to identify the chlorophyll pigment. So scientists have already done many experiments and they have given RF values for photosynthetic pigments. The values depends on the solvent and the extraction chemical. So remember this, RF value depends on three things. First, the species, the type of plant. Second, the chemical used for extraction. Third, the solvent used for chromatography. Now let's look at the example in your book. Here the species is spinach. The chemical used for extraction is hexane and the solvent is a mixture of petroleum ether, propanone and chloroform in a 3 is to 1 is to 1 ratio. And the stationary phase is silica gel. So based on this, you will be given RF values. So next time you do this experiment, check the RF values, then you can identify the correct chlorophyll pigment or the correct photosynthetic pigment. Question from October 2021. Let's get started. The question says chloroplast pigments can be separated and identified by their RF values. Which process can be used to separate chloroplast pigments? And the options are chromatography, dendrochronology, osmosis, and finally it's PCR. So let's go through them one by one, and chromatography is the correct answer. It's the process used to separate different pigments based on their solubility and movement. And then dendrochronology, that's the study of tree growth rings used to determine the age of trees, not pigments. Osmosis, this is the movement of free water molecules from a region of high water potential to a region of low water potential through a selectively permeable membrane. And finally, PCR, or a polymerase chain reaction, that's a process that amplifies DNA molecules, not pigments. So the correct answer is A, chromatography. So the next question shows a paper after chromatography. You can see the solvent front, the direction of movement, and the spots for pigment J and K. And the question asks, what's the RF value for chloroplast pigment J? As we have learned, we are going to apply the RF value formula. RF is equals to distance traveled by solute over distance traveled by solvent. So first I will measure the distance traveled by the solute from the origin to J, which is 6 centimeter. Then if you look at the solvent front, its distance is 7.5 centimeter. Now we divide 6 by 7.5. It's equals to 0.800. So the correct answer is B. Question is from October 2021, paper 6. Actually, I'm not going to explain this question here because I've already covered how to write a discuss type question in one of my previous videos. If you want, I'll link that video in description box so you can check it out for the full explanation. The next question is from the January 2023 paper. In this question, you are given a chromatography paper with a diagram showing a spotted pigment, the origin line, and the direction of movement of the solvent front. The question asks, describe the measurements taken and how they can be used to calculate the RF value for this pigment. So this is quite similar to what we did in the previous question where we applied the RF value, but here you just need to describe the steps what you would do. The answer is simple. First, you need to measure the distance moved by the solvent from the origin and the distance moved by the pigment from the origin. Then apply these measurements to the equation. RF equals to distance moved by pigment divided by distance moved by solvent. And then by substituting the measured distance into this equation, you can calculate the RF value for the pigment. The next question is from the 2023 June 3 paper. This question is a little bit different because we have already seen chromatography with chlorophyll pigments, but this time they have asked about antibiotics. So how can you apply chromatography to antibiotics? So these antibiotics you are going to study in chapter 6, but here I have brought this question under chromatography. Basically, the question is about antibiotics tetracycline and vancomycin, and they have performed chromatography using two methods, method 1 and method 2, and they have given the RF values for each antibiotics from each method. And the next information is a diagram that shows a chromatogram for one of these antibiotics using one of the methods. This picture is familiar to us. It clearly shows paper chromatography. Be careful with the title at the top. They have mentioned that it's used in one of the methods. Because the following questions are about method 1 and method 2. That's why I'm emphasizing this point here. And finally, the question is describe how this chromatogram could be produced. To do this, if you can remember the chromatography steps, we are going to use the same basic steps here. That's why I told you in my previous video, in one of the chromatography videos, that chromatography is not something unimportant. It's actually essential because in the practical exam, we are not going to perform chromatography. So you have a high chance of getting this type of question in one of your past papers, right? In this question, you can see they are practically testing how you would perform the steps. The practical steps you have to write. It's very simple, just like we did with chlorophyll pigments. So first you have to grind the two antibiotics, put them into a mortar and pestle, and using an appropriate solvent, make a paste. Then using a pipette, you are going to prepare your filter paper. The paper should have a pencil line where you will place a drop of your solution. Next, you prepare your extraction solvent, that mobile solvent, pour it into a beaker and then place the filter paper inside, making sure the solvent level is below the pencil line. Allow some time for the chromatography to run. After some time, you will see the solvent front and the antibiotic movement. The distance moved by the antibiotic and the distance moved by the solvent front are very important, because finally using the RF value we can identify the antibiotic. So this is how we perform this chromatography. So to summarize, the antibiotics are first dissolved in a solvent. It can be water or alcohol. The solution containing the antibiotics is carefully loaded onto a strip of filter paper just above the origin line. The filter paper is then dipped into the solvent below the origin line, allowing the solvent to rise and carry the antibiotics along the paper, and the paper is left until the leading edge of the solvent front is near the top of the filter paper. And this is the method used to produce this chromatogram. And the next question is a little bit of tricky one. This is why I love biology the most, because in past papers here and there I can see some questions where the students has to think logically to answer. Sometimes you even have to use a bit of mathematical or logical thinking. So this question asks you to identify the antibiotic in the diagram and the method used. When we look at the diagram, you can see the origin, and they have pointed out the movement of the antibiotic and the solvent front. If I just estimate, the movement of the solvent is about 4D and the movement of the antibiotic is about 3D. So if I apply the RF value formula, that's RF equals to 3D by 4D, and it equals to 0.75. When I go back to the table, I can see that 0.75 corresponds to tetracycline using method 2. So the answer should be tetracycline method 2. And the next question is explain why the four values in the table are all different from each other. This question comes a little bit from practical knowledge. If you know the practical steps, then only you can answer it clearly. The first thing is that chromatography depends on several factors. For example, the solvent can be different in method 1 and method 2. And also, tetracycline and vancomycin are two different antibiotics, so they can have different solubilities in each solvent, which could be one reason. And you can also see that tetracycline is more soluble in both solvents than vancomycin, which is why they produce two different RF values. And finally, we can say that they might have used different solid phases. Maybe in one method they use paper chromatography, and in another method they use silica gel or another solid phase. So this can also produce different RF values. The next question is the same as the October 2020 paper. So you can see the same type of question here. They are asking which process can be used to separate chloroplast pigments according to their solubility. It's very clear, it's chromatography, none of the other options. Again, you can see the same order of answers that appeared in 2020 October paper. Now the B part. You can see the students separated the chloroplast pigment found in brown seaweed, red seaweed, and spinach using this method. So simply the student used paper chromatography, and they have given you the chromatograms from the three plants. Now you have to describe how the student could identify pigment P using this method. The question you need to understand, they are asking how you can identify pigment P, not how to perform chromatography. So first you have to understand the question clearly and write the answer appropriately. So the student first has to measure the distance moved by pigment P from the origin and the distance moved by the solvent from the origin, because the only way to identify pigment P is by using the RF value. So using these measurements, you find the RF value for pigment P using the equation RF equals to distance moved by pigment over distance moved by solvent. Then the student must compare this RF value for the pigment with the known RF value in the same solvent to determine the identity of pigment P. That's very important, because you must use the same method, the same solvent, the same extraction solution. Everything must be same. Only then the RF value will match and you can correctly identify the pigment P. I hope this video is clear and useful for you. If you want any other specific part in Unit 4 or if you need any past paper series, please comment below. And don't forget to subscribe to our channel and share it with your friends. Thank you.